Okay, so now I'm recording. Okay, so one of the things I wanted to do is I wanted to amplify this part of the diagram over here. So the next slide amplifies that and shows the structure a little bit more. So here's the nucleic acid, and these and so this could be called the backbone of the nucleic acid, or using our beads on a string analogy, this is a string, and these little beads are called the nucleotides. And so the beads come in 4 different varieties, and I'm going to get to that later, but at this point, I'm just going to say there are 4 different kinds of nucleotides. And in fact, I'm not going to say what they are at this point, but they have letters. And in the nucleic, in the RNA, the nucleotides are, at this point, I'm just going to use letters. They're G, C, U, and A. So consider these as beads. And the beads are distinguished from one another by 4 letters, G, C, U, and A. And so here's an A down here. And the way it's programmed, we have to, I'm going to go back and talk about a computer again, and everything. When computers work, they basically work on a binary method of storing information. And it's either true or false, or if we had the beads, we'd only have 2 color beads, they'd be white or black. And so that would be a binary coding system, which I think we're all familiar with. And so in binary, if you have 1 bead, it can code for 2 different things. If you have 2 beads, then you can code for 4 different things. If you have 3 beads, you can code for 8 different things. You keep multiplying by 2. Now, if you have 4 different color beads, the first bead can code for 4 different things, and the second bead can code for how many do you think if you had just 2 beads? The first could code for 4, and the second one could
16.
Yeah. So for each of the values of the first one, you can have one of the values of the second one. So 2 of these can code for 16. If you have 3 of them, they can code for how many?
64.
Yeah. So it's 4 times 4 times 4, 64. So with this code, then 3 elements, 3 different beads can code for 64 different things. And these nucleotides taken 3 in a row are called a codon. So that's kind of the word. So here's 1 codon, here's the second codon, here's the third codon. And so this codon codes for an amino acid, which at this point we're just going to call A. That's 1 amino acid. This A is different from this A. This A designates one of the 20 amino acids, and this A designates one of the 4 nucleotides. So this codon codes for A. Here's another codon. It codes for an amino acid, which we'll call T. here's a codon, and so on. And there's some special codons that don't actually code for amino acids. And this one, UAG, is called a stop codon. And so basically, when the ribosome manufacturing process comes across this codon, it says stop the manufacturing. That's the end. That's the end of this this translation. Okay, so if you all generally follow that, then I'll go a little further and tell you what all these things are. Did you follow this? The idea that these
Bob, you ought to emphasize that the sequence of the ATE, etc. When you put those together, that gives you the protein.
Yeah, well, that's kind of what I'm trying to show here.
Yeah, but you didn't talk about it.
Okay, well, this line here is basically, these are like beads on a string, and so this is the protein. Now, this string, I'm going to tell you what the string is a little bit later, you know, but this is a primary. This is a protein. Just a string of amino acids is a protein. Let me go just a little to the next slide.
I understand, while mathematically you've got 3 nucleotides
Mm-hmm.
for one codon.
Mm-hmm.
And if I have 3 nucleotides and each one I can choose to be one of 4, that gives me a lot of choices. Now, somehow that gets funneled down to 20 or 21, whatever, for the number of amino acids. So a lot of those choices I could possibly make either don't do anything or don't exist.
Yeah, I'm going to tell you, I'm going to get into that later.
All right.
Well, wait a minute. Just a separate codon can code for the same amino acid. For example, alanine. Let's say A is alanine. Alanine can be coded by not only GCU, but it can also be coded by some other triplet. Sometimes 3 triplets can code for the same amino acid.
So that's how the funneling takes place, I
Yeah.
get. Okay.
Yeah, that's part of the reason. The other reason is the stop codons.
Yeah, I'm going to actually show you a table that translates the codons into the amino acids.
What about the order of the codons? Does that matter? Does that change the nature of the resultant protein?
Oh, sure. Just like a binary number. If you have 1, 0, 0, that's different than 0, 0, 1.
Okay.
Well, I mean, it's, you know, the order matters. And so this is the first, the order does matter. And in fact, when you start translating, you have to be very careful that you start in the right place. And there's a lot of mechanisms to make sure that you start translating in the same place. If in fact, instead of starting with this nucleotide, you started with the next one, then this would be the codon. And so that would be a frame shift error. And so you just get nonsense. If somehow it missed one and you shifted the frame, then what you got coming out of it would be nonsense. And that's called a frame shift error. But the body has all kinds of mechanisms. Animals have all kinds of, and plants have all kinds of mechanisms to detect these errors and repair them, you know, which we're not going to get into. Okay, so moving along, the next thing I want to do is talk about what the building blocks are. And first I'm going to talk about the building blocks of RNA and DNA. And then I'm going to talk about what the building blocks of a protein are. But so here's the building block of this one happens to be a building block of DNA. No, this is a building block. No, this is a building block of RNA, it turns out. And I'll get to that a little bit later. But in this case, there's three elements to the building block. And they call this the nitrogen base in this. This whole structure in fact, I have to go back to what Jessica just said. This whole structure is called a nucleotide. And this, it's got three parts. This is the base. Then there's a sugar with 5 carbons. It's a 5-carbon sugar. And then there's a phosphate group on it. And so this is just a kind of a block diagram here. Here's a chemical symbol. So this is one of the sugars. And this particular sugar, I'm sorry, this is one of the bases. And I think the nomenclature that I'm going to use it's in one of the following slides, or in fact, let's see if it's.
You had no mail.
The nomenclature I'm going to use for these little elements is nucleobases. There's various nomenclatures that you can use, but did I talk about this slide? I think I jumped ahead. Let me go back to, well, we're going to call these little building blocks the nucleobases. And when I said that there was C, G, A, and T for DNA, so this is, the C stands for cysteine, the G stands for guanine, the A stands for adenine, and the T stands for thymine. And so these are the chemical structures of these nucleobases. And for RNA, you have the same nucleobases except for this last one. And instead of having thymine, you have uracil here. So the 4 letters used for DNA are C, G, A, and T. And for RNA, they're C, G, A, and U. And if you look at this diagram here, you see this backbone here. And this is made up of the sugar and the phosphate groups. And here's another backbone. And they correctly made this backbone a little different color than this backbone because it's just a little bit different. And in fact, the sugar used in the backbone of RNA is a little different than the sugar used in the backbone of DNA. So I'm going to kind of go to the next. So here's one of those building blocks, which we were talking about over here. And this is a nucleobase. This is a sugar that has 5 carbons. And in this case, this sugar is called ribose. And this is a sugar that you find in RNA. And then the word DNA is, the word DNA means it comes from deoxyribose nucleic acid. And what does the deoxy mean? And what the deoxy means is that this hydroxyl group here is replaced by a hydrogen. So if you look at this deoxyribose would be called the sugar used in DNA. And instead of having an OH here, it has just a hydrogen. That's the only difference in the sugar. And that's why DNA is actually a longer word. It means that you don't have this oxygen over here. The other thing, is everyone kind of with me so far?
Yeah.
Okay.
I did want to ask something. It might be a little premature. The process, apparently, when the DNA splits in half and creates an RNA out of one half, the RNA changes in character, both in sugar and in that fourth element, which thiamine to something starting with a U. I don't have. What is the process that causes that change?
It's just that the building blocks, the single strand of DNA is a template, and it's just used as a template to construct the RNA. But the building blocks of RNA are slightly different. And
I understand that, and I'm wondering,
but
what causes it to be different?
Oh,
well, there's a couple of things. The first thing is that that Bob has not talked about is there's two types of RNA. There's messenger RNA and there is transfer RNA. Transfer RNA is very short and it lives in the cell all the time and is what you actually make the small acids that you build proteins from. The other one is used to make a duplicate of the information from the DNA. So I hope that helped.
Can I interrupt for a moment here? Maybe I can help you, Ligel. If you have a code in the DNA, let's say the code is ATGC, then when the RNA copy is made, wherever you have an A in the DNA chain, you have a U in the RNA chain. Wherever you have a G in the DNA chain, you have a C in the RNA chain. In other words, for every nucleotide in the DNA, there is a specific nucleotide that is placed in the RNA. So the code message, the information in the DNA is translated that way into what Bob mentioned, the messenger RNA, and that messenger RNA carrying the message of the DNA then leaves the DNA and goes out to the ribosome where its message in turn is translated into a specific protein. Does that help?
It does, but I don't think it addresses my underlying question. However, my question might be a little more than we want to go into in this lecture, so I'll pass on it.
What was the other RNA called?
Pardon?
Oh, there's several different kinds of RNA. One is called, the one Bob talked about was transfer RNA.
And the messenger.
Messenger is a
I presume they're constructed the same way?
Yeah.
Well, not exactly.
Well, go ahead, Bob, if you want.
I mean, the transfer RNA is already sitting in the cell, and it is the messenger RNA that tells the transfer RNA to make the amino acids and build the protein. So it's a multi-step process, but the transfer RNA already exists in the cell, and you use the messenger RNA to link the pieces that are already in the cell. Okay.
I'm just going to make one comment on your statement, Bob. Is the amino acids, talk about what an amino acid is. The amino acids are in the foods we eat. Some of the amino acids are in the foods we eat. And I would say that all except 9 are, well, let me see. Yeah, all except 9 can be manufactured actually by the body, but there are 9 amino acids which are called essential amino acids. And the body can't make these amino acids. So you have to get them in the food you eat. And that's why vegetarians, they have to be careful because there's certain amino acids that beans and certain plants don't have. So they have to make sure that they supplement their diet with something so that they can get these essential amino acids. And the other thing about amino acids, you can go to your local health food store and buy them. You know, if you go to the shelf, you'll see all kinds of bottles containing, you know, these amino acids. And so you can buy these things and eat them and to supplement your diet. Okay, let's see, did I finish? Now, and so this is a phosphate group, and I guess I don't say it here, but I think I do actually. I will have a slide that actually defines how you name this. I'm going to call this a nucleobase. This is a sugar, this is a phosphate group. When you have this base and just the sugar, it's called a nucleoside. And when you have this base, this sugar, and a phosphate group, it's called a nucleotide. And it turns out that if I gave you all these names, you'd increase your vocabulary by 10%. You know, there's so many words. And so if this nucleobase were adenine, then this base plus this sugar would be called adenosine. And I think this base plus this sugar plus this phosphor would be called adenylate or something like that. But we're not going to use these words. There's another, for the future, there's another point I want to make here, and it's not shown on this slide, but you see references to it a lot later, is that in this sugar, they number the carbons. And when they number the carbons, this carbon here is called the 1' carbon. This is 2', this is 3', this is 4', and this is 5'. And so you're going to hear in later lectures where they're going to talk about say the 3' end of a nucleic acid or the 5' end of a nucleic acid. And the reason they call these primes is the carbons in the base are numbered and they're not primed. And so this is 1', that's where the base attaches. This is 2', that's the one that either has a hydrogen or a hydroxyl group on it. This is 3', and this is one of the points where linkage occurs. This is 4', and this one is called 5'. Okay, so let me talk about the linkage just a little bit more. So if you can see where my pointer is, this is a previous slide. So this is the same combination of a base, a sugar, and a phosphate group, and this is called a nucleotide, the 3 of them together. And in this point, the base is called, this is cytosine, and they're calling it a nitrogenous base here also. So, but it's just the base. And here's the sugar, and then here's the phosphate. Now, the question is, how does this replication work? And so this slide illustrates that. And so if you look on the top here, you can see this strand of, in this case, this is DNA because they have a T here instead of a U. So this strand is the template. And if you can see, it's upside down from this one, you know, and so we talked about the sugar. So this is the 3' sugar. And so if you look from here, this is 1, 2, 3. So this would be the 3' end of the template. And this one here is the 5' end. This is where the 5' end of the template is. So when they talk about replication, they say that the template is read from the 3', it's read from the 3' end to the 5' end. So when you start constructing the new strand, you start up here at the 3' end as a template. But if you look at, we'll call this one a complementary strand. So this carbon here is the 5' end. So the complementary strand grows from the 5' end to the 3' end. So this is read from the 3' to 5'. It grows from the 5' to the 3' end. That's, you may not remember that, but that's kind of, later on, people will be using those words all the time. And so how does this strand grow? Well, you have, first of all, you bring in a nucleotide. You see there's a T over here, you start reading it. And so you have to bring in a nucleotide that matches, that pairs with the T. And it turns out that A pairs with T. And so if you see a T here, you have to bring in a nucleotide that has an A on it. And so you put an A here. Then you read the next one, it's got a C. So this one has to have a G. So you add this nucleotide. This one has an A, so you put one with a T, and you add this nucleotide, this one has a G. And so that's the way this chain grows. And yeah, I'm gonna detail that just in the next talk. I'm gonna talk about this a little bit more because that's important, but that's enough for now. Do you have any questions? But you gotta follow how this half of the DNA, one strand acts as a template, and then you grow a complementary strand, and it grows in this fashion. And this is the process. This is the process I will be talking about in the second talk. And when we grow these things, it's called polymerized chain reaction. And that's going to be, we'll talk about this a whole lot more in the next part of the talk. The other thing I want to do is kind of show you do the same thing with proteins. Proteins are a lot easier actually than nucleic acids. And so here's a protein, and this, we talked about this last time. This was the antibody protein. And if you remember the talk, these antigens could be the viruses that invade the body. And as a defense against these antigens, the body produces antibodies. And and they produce antibodies such that this part of the antibody kind of, it's a lock and key with this antigen. And so it can capture these antigens and bring them to a place in the body where they can be destroyed. But I wanted to show you this just as an example of a protein. And this is actually a protein with what they call quaternary structures. This protein has 4 different strands. Here's one strand, here's another strand of protein, here's another one, and here's another one. And then these proteins come together to form this complex structure called an antibody. But then the question is, what are the building blocks of proteins? And so this is the building block of a protein. It's kind of much simpler. It's in some respects simpler than the nucleic acid. And it's kind of got a backbone. This is the backbone here. There's a nitrogen, and I think this is called an amine group, but that doesn't matter. And on this end, there's a carbon. And in the central carbon, there's a thing which they designate as R. It's called a side chain. So there are 20 different side chains, and the side chain determines what the amino acid is. And so that's how you distinguish one amino acid from another amino acid, is they all have different side chains. Okay. Now, it turns out I have a slide that shows you how these amino acids bind together to form a protein. And I'll show that. But is there any questions before I do that? Is everyone with me? Anyone with me?
I'd just like a little clarification. Are you saying that each of these circles is an amino acid and
No.
together they are forming a protein?
No, I'm saying the whole structure is an amino acid. And this is a carbon atom. This is a carbon atom. I'm sorry, this is an oxygen atom, hydrogen atom.
So
Okay.
we're looking at an amino acid right here, not a protein.
No, this is just an amino acid.
And would you just explain again what the R is?
It's a side chain. So this is a long, complicated molecular structure that distinguishes one amino acid from another amino acid. I said there were 20 different amino acids that we could code for. And so this is a side chain.
Okay.
And every amino acid has a different side chain. So, and I think the next slide, they'll show how the amino acids bind together, and they will label the different amino acids with an R1, R2, R3, R4. You know, that's what they'll put down here. But the other thing I could have done, instead of putting an R here, I could have put the letter. There's 20 different amino acids and they all have a letter. I could have put the letter of the amino acid here. So let's go on and see what happens on how they. Okay, so let me move this over so we can. So this is how the bond works. How this is one amino acid, and you see this amino acid is same as this amino acid, and this one is called R1, and here's a different amino acid, and the side chain is called R2. And so this they call the N-terminus because it's got a nitrogen here. This is a C-terminus because it's got a C here. And when they talk about a protein, they can talk about the N-terminus and the C-terminus. It's the same as I talked about with amino acids. I said I distinguished between one and the other. One was 3' and the other was 5'. So this is how they do that with an amino acid. And so they bond together, and this bond is called a peptide bond. And you can see what happens. I think you can kind of see what happens. You have two amino acids, and when they bind together, this nitrogen is directly connected to this carbon. So this link here connects directly to the nitrogen over here. And what's left over is one oxygen and one hydrogen here and another hydrogen here. So if you have an oxygen, so here's an oxygen with two hydrogens. And does anyone know what molecule consists of oxygen and two hydrogens? Most of us know that.
Water.
Water. So here's our good old H2O. So when you form this bond, you form the bond here, and you kick out a water molecule for each bond. So if you wanted, in fact, just a comment, if you wanted to break this bond, that would say you hydrolyze this bond. That means you have to add in water to these. To go from here back to here, you have to add extra water in here to break this bond and break the protein down into peptides. And just another piece of nomenclature, when you have two or more amino acids put together, but they're not long enough to be called a protein, they call them peptides. So if I had 5 amino acids, that would be a peptide with, I don't know if there's a special word for it, but if there's 2 amino acids, it's called a dipeptide. And if there's several amino acids, it's called a polypeptide. And so the word polypeptide are little short segments of amino acids, and they're not long enough to be considered a protein at that point. So they might use the word polypeptide. And the same thing happens with nucleotides. If you have little short sections of a nucleotide, only say 2 or 3 nucleic acids, they may call that a, well, I'm not going to say that because the word's already used. Okay, this is a peptide, and this is a dipeptide. And they do use the word polynucleotide to mean, several linked nucleic acids, not just several linked nucleotides, not a whole long chain called a nucleic acid. Okay, so are you kind of see how this works? And well, I'm going to close this now. And now I'm going to kind of come to the finale here, almost the finale. And I'm going to zoom in on this one a little bit so you can read it. Okay, so the question was, there's 4 things here, and so these 4 things are the nucleobases. This is we're gonna talk about translation here, and the question was, what set of codons code for which particular amino acids? And we said since there were 4 different codons, you could code for 64 different things. And this table tells you what each of the codons codes for. So here's how you read this table. So this is the first codon is a T. So if the first codon is a T and the second codon is a T, then you have everything. Then you have everything over here. Now, if the third codon, then the third codon, which is the third base over here, you can see the third. I'm sorry, I'm using the word. This is the first nucleotide, second nucleotide, third nucleotide, and the 3 of them together are called a codon. And And so then this is the third nucleotide. So if you have TT and then you come over here, TTT, it codes for phenylalanine, and so does TTC. So here's a case where two codons code for the same amino acid, which is phenylalanine, and it's designated with the letter F. If you come down a little further, you have a TTA, a TTG. All of these code for leucine, which has got the letter L. So the question is, what do all these things code for? And so you can come to this table and read here what amino acid corresponds to each codon, each sequence of nucleobases, each three nucleotides in this case. And then we said there's a stop codon, and so here are these code for stop the translation. Does everyone kind of understand this table? So if you want it, that's kind of the answer to the question, what are these different, we have some redundancy in the codons. And so here's the redundancy. And, you can ask questions, why redundancy? And, there's all kinds of, it's not as simple as this when you really get into the weeds, but this is the basic idea. Okay, we're near the end. Any questions? Okay, so the last slide here is basically, instead of talking for the, this is the general central dogma. And the primary one was you have DNA, and I think, and DNA can duplicate itself. And we kind of talked how DNA duplicates itself. And there's an enzyme called DNA polymerase that is active in the duplication. And then the DNA can be, I shouldn't say, instead of saying duplicate, I should have said replicate itself. It can replicate itself. You can see that up here. And then you can transcribe the DNA into an RNA molecule. And then you can then translate the RNA into a protein. That's what we just talked about. Later on, they found that life is not this simple. There's a few other things that happen. And if you have an RNA molecule, there's such a thing, an RNA molecule can actually serve as a template to form a DNA molecule. And the enzyme in that case is called reverse transcriptase. And so that's the green line that's here. So if you have an RNA molecule, you can actually form a DNA molecule from it. The other thing they found, that RNA can actually duplicate itself. They never thought it could, but it could. And so this shows that this is called RNA replication. And so you take one strand of RNA and then it can replicate into a complementary strand of RNA, which is shown over here. So that they say this is plus sense RNA, this is negative sense RNA. And then this strand of RNA can be replicated back into another positive sense RNA. But this is the way the coronavirus works. The coronavirus doesn't have DNA in it. It has RNA, And this RNA is used to build all the structure. It's translated into protein, which is all of the structure that consists of the virus. And then it can even make some more RNA. So this is basically the way that the coronavirus works. Now, the way we test for the coronavirus is we want to see if this coronavirus RNA is there. So we don't know how to duplicate. We don't know how to do this process, but we certainly know how to do this process very well. And this process is called PCR. And so what we first have to do is when you do a nose swab and say you get some of these viruses, then you kind of destroy them to get the RNA out of them. And then you put them into a mechanism. You put some reverse transcriptase in, and then you form DNA. And then we duplicate this DNA with a process we call PCR. And this is what the next talk is going to be all about, is this process over here. Okay, we're done here. So any final questions?
I wanted to ask about the RNA is composed, the coronavirus is made up of essentially RNA, not DNA. And I guess what I wanted to talk about is, from my understanding, it is not considered life or alive because it cannot live on its own.
Yeah, no virus can live on its own.
So a virus is not considered to be life or a form of life, even though it has a structure in it that is part of living things that other living things have.
A virus can't replicate itself.
So is that the definition of life? You must be able to replicate yourself.
Yeah, I would think so. I mean, other various...
Can I interrupt here? Yes,
Sure.
indeed, Miguel. The definition of life is the ability to replicate. And since viruses can replicate inside another cell, they are still considered alive, and they evolved from living creatures, so they are alive.
Yeah, I was going to say your definition of life. They're alive.
Yeah, they're symbiotic.
Yeah.
Okay, I guess I read another definition of life is that it must be able to live, replicate itself on its own, not as a, in effect, well, I guess it's like a almost parasitic in a way. It must have another, invade a cell in order to replicate itself.
Right. Yeah. It's. I think a lot of people have debated what the definition of life is. So let me look at my watch. Okay, so we used a little bit more than an hour. So we're going to have to. We're going to talk about the polymerase chain reaction, and we're going to do the same thing with the next talk as with this talk. I'm going to show some basically YouTube movies is basically what the next part of this is all about. And the first movie is actually, I would consider an excellent movie that basically tells you 80% of what you need to know. So if we get through that first movie, we'll know 80% of what we need to know. And then the remaining time will be used filling in the remaining 20%. So
I just want to ask you a quick question before you take that slide away. If I understand what you have said, you start out with the viral RNA.
right,
You reverse transpose it, using reverse transcription, you generate some DNA, which then replicates itself.
well,
And the idea then is to build up a certain critical mass of DNA that you can detect in some way, or am I missing a step? Or
No, you're right. You're right, actually. It turns out that if you do a swab and it has, say, 1 or 2 or 3 strands of RNA, that's not enough to detect. You just, there's nothing sensitive enough. I'm using sensitive and sensitive enough to detect 1 or 2 or 3 strands of RNA. So you have to do something else so you can detect it. And so what you do is you generate double-strand DNA, and then you go into, it doesn't really replicate itself in this case. We have a process called polymerase chain reaction where we speed up this replication. And so we will go through, typically in polymerase chain reaction, you'll go through probably 35 to 40 cycles of replication. That means if you started with 1, you'd have 2 to the 35 DNA molecules.
Mhm.
And 2 to the 35 DNA molecules you can detect. And there's different ways of, different ways. Just because you have the molecules, that's what PCR is. It tells you have the molecules, but you somehow have to have a mechanism that tells you how many molecules are there. And that means you have to put fluorescent tags. You have to put some kind of tag on it to tell you how many molecules are there. And we're going to talk about some of these tags that we put on it and how we detect the fluorescence. And I don't know if you remember when we talked about antibodies, but we put a enzyme that produced the color change in the media that it was in, and then you could detect how many antibodies were there. And a similar thing we have to go through that mechanism for this. Okay, so I'm going to go on to the next one. Now, okay, I start out with the same slide so that it'd be something familiar. And so what we're going to talk about is polymerase chain reaction. And I won't define it because it'll be defined in some of the following movies that we're going to watch. But one of the things I did, this is for Bob Corson's sake. He's always has this little game where he has various Nobel Prize winners, and yet we have a little game where we narrow down which Nobel Prize winner it was. It's like 20 questions. Was it a woman? And what kind of Nobel Prize? But in the field that we're talking about here, here's some of the significant Nobel Prize winners that I think that we all kind of should know about. And the first person on the list is Linus Pauling. And he was kind of the father of all of this bonding. And Linus Pauling went to Europe at the time that Schrodinger and all of the people involved in quantum mechanics were there. And he studied with them. He learned quantum mechanics very well. And then he came back and says, how do I apply quantum mechanics to the binding of different molecules? And so he worked out how molecules bind with each other. And I showed you how the how a protein was formed. And so he wrote a book, which is just a standard in the business, called The Nature of the Chemical Bond. So he's a very important person. And the other thing about Linus Pauling, which Bob Corson can use as a trick question, which is the only person that ever was the sole winner of a Nobel Prize, the sole winner of 2 Nobel Prizes. And there's only one person that was the sole winner of 2 Nobel Prizes, and that was Linus Pauling. And he won the first prize for the nature of the chemical bond, and the second one he won for a Nobel Peace Prize. And one of the things that many of you may remember Linus Pauling from is that in his later life, I think he died at 90s, he suddenly decided that eating huge amounts of vitamin C was good for you. And so all of us, I think, were taking huge amounts of vitamin C at that time. And the other people that are that discovered the structure of nucleic acid, they worked out the structure and they got a Nobel Prize. Was Francis Crick, James Watson, and Morris Wilkins. And in 1962, they got a Nobel Prize for that structure. The person missing from that list was Rosalind Franklin. And Rosalind Franklin was really a very key contributor. And she was the one that did X-ray refraction patterns. And she had a pattern that showed that the structure had to be a double helix. And once Francis Crick and James Watson understood that from her data, they very quickly put a model together, which showed that, you know, the model of that we've been talking about of the nucleic acid. And the person that showed them the X-ray that Rosalind Franklin was working for Morris Wilson, he's the one that showed her X-ray to Francis Crick and James Watson. And, you know, so there's a very interesting story here. The fact that Rosalind Franklin should have shared the Nobel Prize instead of Morris Wilkins, I think. But you know, by the time the Nobel Prize was awarded in 1962, she had died. And I think she died from breast cancer, and it may have been, you can speculate it was a result of working with X-rays all the time. And there's another, Bob Corson needs some ladies that won Nobel Prizes. There's another Rosalind, and her name is Rosalind Yallow. And when I was at Bio-Rad, she was an important person. You know, there were rooms named after her, the Yallow Room. And she was the lady that basically did the first immunoassay. And instead of tagging with an enzyme, they tagged with a radioactive molecule, and these were called radioimmunoassays. And Bio-Rad sold radioimmunoassays for quite a while. You know, one of the reasons was the word bio in the name. But in fact, I asked some of the old timers at Bio-Rad, you know, where did the word bio come from? And they said, well, it was in their log before the radioimmunoassay was invented. So they were into these radioactive kind of assays for a long time. And the third person is credited with got a Nobel Prize for discovering the polymerase chain reaction, and his name is Kary Mullis. And so we're going to learn all about him. So let me go on. So we've talked about this one. This first movie is I would consider the way movies should be made. This is a very good movie. And so if we get through and understand this movie, we've got the 80% of it. So I'm going to play this movie now.
Is there any sound on this?
Oh. Yeah, I didn't set myself up right, so I got to get out of this. Can you, I got to go all the way back here. I didn't, wait, how do I, when I logged in, I should have.
If you go to screen share, there is a button to push says share computer sound.
Okay. Yeah, that's where I should have done it, yeah. Thanks, you're the czar of Zoom. Knows all the answers.
Yeah, right, yeah.
So now I got to share again. Okay.
Share your screen.
Yeah, I'm doing that, yeah. Okay. So we're going to start all over again.
Using reverse transcription polymerase chain reaction, RT-PCR, in COVID-19 testing. COVID-19 testing usually begins with a swab of the nose or throat. Saliva, mucus, or fluid from a patient's lungs can also be used. The swab of a person with COVID-19 will contain a mixture of human cells, virus particles, and other microbes. Like all living things, the human cell has DNA as the genetic material that passes on information from one generation to the next. The DNA molecule...
I wanted to stop here because this is really an important picture, you know, in terms of understanding. So we were talking about the DNA double helix, but basically here, most of us are familiar with chromosomes. And if you're into genetic genealogy, it's all about chromosomes. And I think we have, the human body, we have 23 chromosome pairs. 22 are the autosomal chromosomes, and there's a pair which is the sex chromosomes, and these are in the nucleus. So this is an important. This ties the double helix into chromosomes, and if you go back, the chromosomes wrap around. Let's see, what are they? Jessica, do you know they wrap around these little proteins, which are spools. Do you know the name of those proteins? It just slipped my mind. Anyway.
I think we lost her.
Anyway, these are little proteins. They wrap around these proteins. These are called histones. And then these proteins kind of form a structure like this, which is called chromatin. And then the chromatin forms a structure like this, which is called a chromosome. And these chromosomes are in the nucleus. So I think this is a useful slide to tie the DNA to a chromosome. So I'll just comment on that. Okay, so we'll continue.
Is made up of two strands that look like a twisted ladder or double helix. However, the COVID-19 virus, SARS-CoV-2, and many other viruses, including HIV, have RNA as their genetic material, genome. RNA is chemically very similar to DNA, but has only a single strand. The virus RNA is surrounded by a nucleocapsid protein within the virus envelope. Other proteins are embedded in the envelope itself. The SARS-CoV-2 genome contains genes, blue arrows, that carry the directions for making these and other proteins that are needed to replicate the virus inside the human cell. The objective of COVID-19 testing is to identify part of the viral genome in the patient's sample.
Okay, I'm going to stop here again. And so this strand of RNA, here's the strand of RNA, and it's a template for making all of the proteins that you saw on the coronavirus. And this protein, I think maybe they talk about it a little bit more, but this is a protein that kind of anchors it to the cell that it's going to invade. These are other proteins. And this is the protein that it typically, when we test for the presence of this RNA, we'll take this little section that codes for the N protein, and this is what is usually amplified.
This is usually the N gene, which carries directions for making the nucleocapsid protein. There is not enough viral RNA to detect directly in the patient's sample, so a process called reverse transcription polymerase chain reaction, RT-PCR, amplifies many copies of a segment of the N gene. Short, single-stranded pieces of DNA, called primers, recognize unique RNA sequences within the viral genome that bracket the target region of the N gene. After the first primer binds, an enzyme called reverse transcriptase extends, synthesizes, a single-stranded DNA copy of the viral RNA, known as complementary DNA, or cDNA. After the RNA is removed, the second primer binds to the other side of the single-stranded cDNA. Then, a second enzyme, taq-DNA polymerase, extends a second strand to produce the double-stranded DNA copy of the target region of the viral RNA. This DNA copy then undergoes successive rounds, cycles of amplification during which the DNA separates, denatures into single strands, both primers bind, anneal, to their target sequences, taq polymerase extends, synthesizes a new DNA strand, and so on. The number of copies of the target region of the viral genome doubles with each cycle. After 30 cycles, up to 1 billion DNA copies of the viral RNA are produced by PCR. In practice, the virus is typically detected with 30 to 45 cycles of PCR. Adding a fluorescent probe allows the amount of target DNA to be detected in real time and quantified after each cycle of PCR. This graph shows.
Okay, so the question is how do you detect how much DNA is in there? And so somehow you have to add a probe to it. And if you started, this green line means there's no virus. And if you started with 2 viruses, they would amplify according to this curve. And the probe would mean that the fluorescence would increase with the number of replications. So the vertical axis is the amount of fluorescence here. And so this is the kind of signal you get out. And so some of the future movies are going to tell what kind of probe you add to get this fluorescence out. So now I'll continue.
The detection of 220 and only 2 virus RNA molecules in a controlled study. Results from swabs vary. A negative result is also shown. See these DNA Learning Center animations.
Okay. So did you all follow that?
I think so.
Yeah, okay. Now,
I got a little lost between, they said they made 1 billion molecules in 30 cycles, and I assume it would have been more than 1 billion. And then we jumped to 2 and 200. I got the disconnect there.
no, if you started with 1 virus and you doubled it 30 times, then you'd have 2 to the 30th viruses, and that's 1 billion. 2 to the 30th is just about 1 billion. 2 to the 10th is 1,000, 2 to the 20th is 1 million, 2 to the 30th is 1 billion. So that's if you had one virus. If you had 2 viruses, then you'd have, instead of 2 to the 30th, you'd have 2 to the 31 because you started with 2. And if you had 4, you'd have 2 to the 32. Are you following what I'm saying?
Are you saying that when they detect this in the nose, they're coming out with a swab with 1 or 2 viruses? You're not saying that.
Yeah, I'm saying that. I'm saying that, yeah.
I find that hard to believe.
Well, it's the way it is, yeah. I'm going to show you, if we get to the end, I'll well, whether it's 1 or 2 or 7 or 8, you know, it doesn't really matter, but it's not very many. And in fact, if it were 1, you could detect it. If 1 is enough, you could take 1 and amplify it to 1 billion. So this is a very sensitive test. Okay, I'll be sending this. I'm not going to show this second movie, and it's also a good movie made by the same group, but it has more to do with using it for genetic engineering rather than what we're doing for testing. So I'm not going to show it, but I'm going to show a short segment of this next movie.
In hindsight, it was such a straightforward idea. What if, instead of using cloning techniques to generate enough DNA for analysis, which takes weeks or even months, you could use temperature cycling and enzymatic replication to generate millions of copies of DNA in a fraction of the time? The year was 1983. Biochemist Kary Mullis was driving late one night when struck with a flash of inspiration. Stopping in the road, he wrote on the back of a receipt the idea that would ultimately win a Nobel Prize in chemistry. The concept, polymerase chain reaction, or PCR, seemed at once elegantly simple and incredibly versatile, a game changer for everything from forensics to diagnosing and monitoring disease. Today, PCR is considered one of the most important scientific advances of the 20th century. Yet, as radically simple an idea as PCR was, it required many pioneering innovations to become the gold standard molecular application it is today. Mullis and his colleagues at CETUS furthered PCR through the 80s and partnered with Roche to develop it for diagnostics. Roche later acquired the patents from CETUS and launched its first FDA-approved PCR test in 1993. Over the years, PCR has been steadily and systematically improved.
So I stopped that movie there. It's a Roche movie, and Roche basically owned the patents to PCR for many years, which they got from CETUS, and sort of delayed the development of PCR and delayed the development of PCR. But anyway, that was that movie. Okay, now I'm gonna skip all of these, and I'm gonna just go straight down. We've seen some of these before. We've seen enough of this, and I'm just gonna go down to kind of how PCR works. And this diagram basically tells you how PCR works. This little device here is called a thermocycler.
Excuse me, could you go full screen with that?
Oh. Yeah, I can make it bigger. Is that okay? Better?
Yes.
Okay.
Can you do it full screen?
I'm kind of reluctant. It really won't get much better. I'm kind of reluctant. It won't get much better. And let me just do it this way. And basically, the heart of PCR is a thermocycler. And thermocycler is nothing more than an exotic oven. And it goes from one temperature to another temperature and stays there for a certain length of time. And so you can see how it happens here. If you start with a strand of DNA, and if you hold it at 95 degrees centigrade, the strand denatures, which means it separates into 2 strands of DNA. And then you lower the temperature to 55 degrees centigrade, and little short strands of DNA, you could even call these things polypeptides, they match up to the corresponding base on these strands. And so this defines where the replication starts. And here's a complementary primer. And so on the other strand, it attaches here. And this is where replication starts. And then if you reduce it to 72 degrees centigrade, then the strands just replicate. And then when the strands have replicated, then you started out with one strand, you have 2 strands, then you repeat the process all over again. And the process is almost as simple as it's shown here, is you take all these PCR components, and in this case, they say there's a DNA sample, but if we don't have a DNA sample, if we have an RNA sample, we could say this is an RNA sample, but we have to add the enzyme reverse transcriptase to this mix. So when you, during the first cycle, there's reverse transcriptase there, and that translates the RNA into DNA, and then this cycle continues. So you have to have a reverse transcriptase in there. Then you have to have specific primers, which tell you where you want to start, and basically this tells you where you start. This other primer tells you where you end. And this video, this diagram doesn't show what stops the replication. Other ones will. The one that I missed will show you how that I didn't show you how that stops. Then you have to add all these nucleotides. And in this case, they're special nucleotides, and they have 3 phosphate groups on them instead of just a single phosphate group. And if you take the adenine, is kind of interesting, but adenine with 3 phosphate groups, another word for it is ATP. And ATP, I don't know if you know anything, ATP is the power battery. It stores the energy for which drives all biological reactions. And so by putting the extra phosphates on, this is where you get the energy to drive these reactions. And then you have to have a polymerase called Taq polymerase. Then you have to have a mixed buffer, which is typically supplied by whoever's supplying you the test. And then you have to put them all in a tube, and then you put them in the servocycler and leave it there for however long 30 cycles take. And when you bring it out, you have 1 billion copies of the DNA sample that you put in there. Taq polymerase is a kind of an interesting polymerase. If I would have continued the Roche movie, they would have talked about it. But when they first started, in order to speed up the reaction, you want to go to a very high temperature, like 95 degrees. And the polymerases that we use in our body don't survive at 95 degrees. So you have to keep adding polymerases to this reaction to keep it going. So someone went and found a bacteria that existed in the hot springs in Yellowstone National Park. And it was hot there. They took the polymerase from that bacteria, and that's called Taq polymerase. And that's the polymerase that they use in this reaction because it'll withstand these high temperatures.
This is fascinating. I wanted to ask you the extension, those orange things, you called them poly or?
No, these are primers. They're called. They're just little short segments of DNA that find the complementary pattern over here, and they attach to it, and they start the process. Now, they only show 3 here, but typically there's not 3. There might be 30 or 40. They just, in this illustration, they just show 3. If you make it too short, it can attach to the wrong place. Go ahead.
These are artificially introduced in the laboratory, is that correct?
Yeah, these are manufactured.
Okay.
And
I'm just trying to, obviously, for this, this process of replication, heat is an element. And I'm just trying to, I am wondering, what is this a similar process that's going on in the human body with the replication of the RNA from the virus?
Yeah, but yeah, when you replicate DNA, or yeah, it's the same process in the body, but it's going along much slower. So you don't have to go at these high temperatures. It just goes along in its own rate. But we want to speed this process up. So we use this PCR process to speed it up. And at high temperatures, this happens much faster. And it might take something like a minute. No, it doesn't take that. Yeah, it could take about a minute per cycle. And for this to happen. Okay, we're going to run out of time. So what I'm going to do is this is kind of clear, is that right? You take all these things and you put, you have to, instead of a DNA sample, you put an RNA sample, and you add some reverse transcriptase into this mix. You put them into a tube. You put this in this thermal cycler, and this thermal cycler generates many, many copies. Like if you had one strand of RNA, you'd end up with 1 billion strands of DNA. And I think I have to show you one more movie. I was going to go to the last movie, but I'm going to have to show you one more. Okay, so let me go here. This is actually, I'm going to show you this movie, and I may skip parts of it. This one, the pronunciation in this movie is terrible. And so.
COVID-19 is an infectious disease caused by severe acute respiratory syndrome coronavirus 2. When a person is infected, the most common symptoms include fever, cough, and shortness of breath. To start a test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx, then it is. Coronavirus testing is polymerase chain reaction, PCR. which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation. To extract the viral RNA by using commercial kit, the sample is first added into a microcentrifuge tube. Then it is mixed with a lysis buffer.
I'm going to skip this. This is kind of
Once the lysis buffer is added,
you can
the procedure is carried out. It consists of a silica matrix, a clean membrane. This removes any remaining. The elution buffer removes the viral RNA. Step, a master mix is used, which is a premixed concentrated solution that consists of buffer, reverse transcriptase enzyme, nucleotides, forward primer, reverse primer, Tachman probe, and DNA polymerase. Finally, to complete this reaction mixture, the RNA template is added. The tube is mixed by pulse vortexing. Then the reaction mixture is loaded into a PCR plate, which generally contains 96 wells, allowing the analysis of several samples at the same time. Next, the plate is placed in a PCR machine, which is essentially a thermal cycler. Real-time RT-PCR is used for the detection of the new coronavirus 2019 by the amplification of target sequences in the RDRP gene, the E gene and the N gene. The choice of the target gene depends on the primers and the probe sequences. The first step in RT-PCR is reverse transcription. The first strand complementary DNA synthesis is primed with the PCR reverse primer, which hybridizes to a complementary part of the virus RNA genome. Reverse transcriptase then adds DNA nucleotides onto the 3' end of the primer, synthesizing DNA complementary of the viral RNA. The temperature and duration of this step depend on the primer, the target RNA and the reverse transcriptase used. Next, an initial denaturation step is applied, causing denaturation of the RNA-DNA hybrids. This step is required for the activation of DNA polymerase and simultaneously the inactivation of reverse transcriptase. PCR consists of a series of thermal cycles, with each cycle consisting of denaturation, annealing, and extension steps. Denaturation step consists of heating the reaction chamber to 95 degrees Celsius, and it is used for denaturation of the double-stranded DNA template. In the next step, the reaction temperature is lowered to 58 degrees Celsius, allowing annealing of the forward primer to its complementary part of the single-stranded DNA template. The annealing temperature relies directly on length and composition of the primers. In the extension step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding free nucleotides from the reaction mixture that are complementary to the template in the 5' to 3' direction. The temperature at this step depends on the DNA polymerase used. After the first cycle, the double-stranded DNA target is obtained. Then, the denaturation of this double-stranded DNA is performed, yielding 2 single-stranded DNA molecules. In the next step, the reaction temperature is lowered, allowing annealing of the primers to each of the single-stranded DNA templates, and annealing of the Tachman probe to its complementary part of the target DNA. Tachman probe consists of a fluorophore covalently attached to the 5' end of the oligonucleotide probe. The fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, this probe consists of a quencher at the 3' end. The close proximity of the reporter to the quencher prevents detection of its fluorescence. In the extension step, DNA polymerase synthesizes new strands. When the polymerase reaches a Tachman probe, its endogenous 5' nuclease activity cleaves the probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released, resulting in an increase in fluorescence intensity, proportional to the amount of amplicon synthesized. This method allows the estimation of the amount of a given sequence present in a sample. The number of double-stranded DNA pieces is doubled in each cycle, therefore, PCR can be used to analyze extremely small amounts of sample. For the measurement of the fluorescence signal, a tungsten halogen lamp, an excitation filter, mirrors, lens, an emission filter.
Okay, since we're running out of time, but I just want to show you one advanced form of PCR, which I think Bio-Rad pretty well owns the patents on, and this is one of the, it's an extremely sensitive method, and there's some other videos here you can watch when you get the time, but I do want to show you this one. So let me just kind of show you this last video.
The human genome is of extraordinary complexity, with its 3 billions of DNA-based pairs that form the thousands of genes that make up our biology. Understanding this complexity requires powerful tools. Digital PCR is a powerful new tool for quantitative DNA measurements. Like real-time PCR, digital PCR uses a simple sample preparation that utilizes TaqMan primers and probes and an optimized supermix. Real-time PCR provides relative quantification. In contrast, digital PCR provides absolute quantification of target DNA sequences in the sample. Let's see how droplet digital PCR achieves precise DNA measurements. The key is sample partitioning. With droplet digital PCR, reaction mixes are partitioned into approximately 20,000 evenly sized droplets, each 1 nanoliter in volume. PCR is performed on each individual droplet. If a droplet contains the target sequence, it will be amplified and a TaqMan reporter dye will emit a fluorescent signal. If the target sequence is not present in a droplet, no PCR will take place and only weak residual fluorescence is emitted. By scoring each individual droplet for a fluorescent signal, droplet digital PCR achieves absolute quantification with a high level of precision and single molecule sensitivity. To generate digital results, samples are amplified to endpoint in a conventional thermal cycler. Droplets that contain the target DNA sequence are read as positive. Those that do not are read as negative. The fraction of positive droplets determines the concentration of the target DNA sequence in the sample. Now let's look at how the Bio-Rad QX100 droplet digital PCR system performs digital PCR with unrivaled precision and sensitivity. The QX100 system uses a simple workflow that requires minimum hands-on time and procedures that are easy to perform. First, 20 microliters each of 8 prepared samples are transferred to the droplet generation cartridge. Droplet generation oil is then loaded into the corresponding channels of the cartridge. There are 8 oil channels, 1 for each of the samples. Once the cartridge is loaded with prepared samples and oil, it is placed in the QX100 droplet generator. Using microfluidic technology, the droplet generator combines the samples and oil to create an emulsion of approximately...
This is what I wanted to show you here. The sample that they prepared comes in here. And in fact, there were 2 oil wells, one here and one here. She said we just fill one, but you fill both. And so oil comes in these channels, and this is on a microstructure. And what happens is they apply a vacuum over here. So the vacuum sucks the sample and the oil, and they mix here. And the oil forms little droplets, and each droplet is about a nanoliter in size. So you typically, you form 10,000 different droplets. So when they talk about a vial in the previous ways of doing PCR, each droplet is a vial. And so then they subject all of these droplets to PCR, and then they check to see which one had one or more virus DNAs in it. So I'll let this proceed.
20,000 mono-sized droplets for each sample. Every droplet contains a nanoliter volume of the sample, including any target sequences that may be present. After the droplets are generated, the emulsified samples are transferred to a standard 96-well PCR plate using a standard 8-channel pipette. The plate is then loaded onto a thermal cycler where PCR is performed to amplify the target DNA to endpoint. When PCR is complete, the plate is loaded onto the QX100 droplet reader. The QX100 droplet reader is an automated liquid handling device equipped with optimized microfluidics designed for precise, reproducible detection of target DNA sequences, droplet by droplet. The droplet reader automatically sips each sample, singulates all the droplets from a sample, and then streams them in a single file past a 2-color fluorescence.
So basically what it does, it puts the mixtures of droplets into the droplet reader, and then, as she said, streams it by a fluorescent detector. Now, if there was one or more viral particles in the droplet, you'll get a signal. If there weren't, you won't get a signal. So basically what they do is they say how many droplets had a signal and how many don't have a signal. And by counting the number that don't have a signal, they basically can apply Poisson statistics and determine what the average density of the virals were in the sample. So I'll just let that continue.
Detector. The detector reads each droplet in turn at the rate of 1,000 droplets per second and determines which droplets emit fluorescence and which do not. As each droplet is read, the data are recorded graphically by Quantisoft software. The software records the positive and negative droplets in each sample.
Okay, did you follow that at all? So what they do is they have 10,000, each sample is partitioned into 10,000 different droplets. And then they subject that mixture of all these 10,000 droplets to PCR. And if there was one or more viral particles in the droplet, then you get a signal out. And if there were no viral particles in there, you don't get a signal out. And then by counting the number of droplets that had a signal, you can determine what the average density of viruses were in the original sample. And I put a little bit of arithmetic here. I showed a Poisson distribution, and and so N is the number of viruses in a droplet. So this, and A is the average number of viruses per droplet, and PRN is the fraction of droplets that contain a virus. And if you look at the number of droplets that don't contain a virus, then N is 0. So A to the 0 is 1. N factorial is 1. So the fraction that don't contain a virus is just E to the minus A, where A is the average fraction. And you reverse this, and then you can get the density of viruses in the original sample. Did anyone follow that?
Yeah.
Anyone?
More or less, yeah.
So that's the method that is extremely sensitive. And the videos that I left out are the way it is probably currently done. And what they do is they put the vial in PCR, and while the sample is being multiplied, they look at the increase in fluorescence. and they have to have a calibration curve. And by monitoring how fast the increase in fluorescence is changing, they can get an idea of the density of viruses in the original sample. So you get a curve. The fluorescence increases intensity with replicate cycles, and that's the way they do this. That's regular, what they call quantitative PCR or real-time PCR. Well, that's real-time PCR, and this is another form of quantitative PCR. And there is a form just called digital PCR, where in fact, instead of using oil droplets, they use little micro vessels, and I think they get up to 10,000 actual micro vessels, in which that's called digital PCR. But when the vessels are these oil droplets, that's called digital droplet PCR, and that's actually one of the products that Bio-Rad has. And the video that I showed was a video from, I think, about 2012. And so they were actively developing that when I was there. And ever since then, what they've been doing is using this method, trying to find applications that need this kind of sensitivity. And it's used more in looking for cancer, little DNA segments that might predict that you're going to have cancer. So there's a lot of publications coming out. And so I think for the last 7 or 8 years, they've just been giving these to research institutes, and they're trying to find applications where only this technique will work and others won't work. But they do have it out. It is one of the tests that's authorized by the CDC under the Provisional Authorization Act for detecting for the coronavirus. But it's probably a little bit more exotic and sensitive than is needed. Okay. Any questions?
The TAC, well, they're calling it TACMan. You called it TAC something else, but that's the one that comes from the thermal area in Yellowstone, right?
Yeah, the T. If you look at some of the other videos that I couldn't get to, in fact, if you look at the continuation of the Roche video, they'll tell you about TACMan, and it's a polymerase, but the normal polymerases are destroyed at that high temperature that they use for PCR. And
well, is that the one that contains the fluorescence?
No, that's I just forgot the name of that. That's a probe. I forgot the name of that probe, actually. Slipped my mind. No, that's a little piece of it's a polypeptide, and when I told you a short segment of a protein is called a polypeptide, and that's a polypeptide. I forget, it's called, I could go back and see what they called it, but I forget the name of that probe. But it's another thing that you need. You need a little probe that when they duplicate the strand, they separate the quencher from the part that fluoresces. And if it fluoresces when it's attached to the DNA, the fluorescence is quenched. But when it's knocked off into the solution, then you get an increase in fluorescence. Okay, so next week, Bob, are you going to take?
Yeah, I'll be happy to continue the thermodynamics and hopefully finish up with Maxwell's material and start on information theory.
Okay, so I'll send these out. I probably won't send out the video, but I'll look at it to see. I'll look at it myself to see, you know, how the presentation went.
This was great, Bob. It was absolutely fascinating.
Okay, well, thanks, Miguel. Okay, so next week you will send out the invitation, Bob.
Yeah, it'll be the same one that I've been using all along, but I'll resend it.